Afternoon and good morning, everybody. This is Michael Yang. I'm the CEO and president of ViaCyte, and it's my pleasure to give you an update on our progress here at the company and what we're doing at ViaCyte. Today, where we sit with ViaCyte, we use stem cell technology and novel delivery approaches to address multiple important disease areas. Our lead area that we're focused on is type 1 diabetes, where we're looking to provide a functional cure for type 1 diabetes. We'll get more into that program as the presentation unfolds. Our broader long-term vision, of course, is to leverage this stem cell technology that we have and the delivery abilities to address diseases in other areas like thyroid or liver, perhaps. We are really fortunate to have a group of investors who sit side by side with us, Bain, notably, RA Capital, TPG, and we're enhanced with our work with world-class partnerships. On the cellular side, we work with CRISPR. They do gene editing, where we leverage that gene editing technology to develop a immune-evasive cell line that can be delivered in our delivery approaches. And we work with W.L. Gore. They're the world leader in biopharmaceutical membranes. That helps us optimize our devices. As I mentioned, our expertise is in cells and delivery. And on the stem cell side, as you can see on the left-hand slide, bottom left, what we do is we have well over a decade of experience with know-how and an exceptionally robust IP portfolio that allows us to fully differentiate from a pluripotent stem cell across the various cell stages all the way through the pancreatic type cells, progenitor, precursor, and fully pancreatic cells that produce insulin. And that expertise and that process of differentiation is what we leverage to put into our devices on the right-hand side. You can see an example of one of such devices. We have two device approaches. One is an open device that allows direct vascularization. And as we are in the clinic with this device and with this approach, that today requires immune evasion with immune suppression medications. But in the future, this would be an approach where we've used the CRISPR cells to evade the immune system and obviate the need for immune suppression. We also have a closed approach, an encapsulated device that we leverage with a Gore material that relies on surface diffusion to have membrane transport of oxygen materials, hormones, and etc. They're very elegant design. There's no moving parts. They're slimmer than a credit card and about the size of a Band-Aid. All along, ViaCyte has been optimizing the placement of these devices, the mount of them, the design of them, to optimize for the fully maximal therapeutic benefit. As I mentioned, our initial focus is diabetes, type 1 diabetes. Type 1 diabetes is the smaller of the market oftentimes described, but no less important in regards to the impact it has on patients and the need for innovation. About 1.3 million patients have diabetes, 40,000 are diagnosed every year. And on the right-hand side, you can see that on a day-to-day, hour-to-hour basis, there's a significant patient burden in managing this disease. There's a precision that's required to manage this disease, and imprecise management leads to downstream healthcare costs and high morbidity and mortality. 8.4% of the patients actually have a high morbidity due to hypoglycemic events. And of course, there's billions of dollars in healthcare costs that our attempt and our goal of creating a functional cure would disintermediate these expenses and hopefully provide a better experience for these patients and less morbidity and mortality. When we get into our product and our programs, we measure our success across three different parameters and three different time horizons. Early on, we're able to see at the cell level through histology and staining the ability for us to see if our cells are differentiating into both beta cells that produce insulin and alpha cells that produce glucagon. We also track biomarkers then once to see if the cells are engrafting. We track a biomarker for insulin called C-peptide. And of course, we're going to measure the impact of that insulin that's produced on hemoglobin or A1C. But ultimately, the final outcomes that we're looking for are clinical, meaningful impacts, things that are tracked and endorsed by the ADA, time in range, for example, the amount of time patients spend in range, the reduction of hypoglycemic events, and the reduction of exogenous insulin. Give you some perspective on why we're pursuing this as a goal. Today, on the left-hand side, you can see patients before treatment have quite a variable amount or measurement of their blood glucose. If You can see on the slide on the left, there's a gray bar. That gray bar represents the approved time in range for diabetics or any patient. It's 70 to 180 milligrams per deciliter of glucose. So you can see that the patients on the left are not in that zone very long. On the right-hand side, as you can see, patients seeking innovation from their disease, they can undergo cadaver islet cell transplants. These require immune suppression. There's obviously a limited supply, and actually they're not fully approved here in the U.S., but there is a very good outcome if you can get these to survive and thrive, and the patients are normalized, if you will, inside the time and range. In the middle, there has been innovation for the patients. Best medical devices, continuous glucose monitors and insulin pumps have provided some relief. These are patients now that are able to sleep well without having to, so to speak, worry about hypoglycemic events. They do not typically provide glucagon. And of course, there is a mechanical element, so they're not naturally physiological. So there's still quite a room for innovation. In fact, upwards of 70%, 60 to 70% of patients do not use insulin pumps. As you can see, for where we are at ViaCyte, we have in the clinic, we have shown proof of concept with both of our approaches. Beginning on the left, you can see that on the open device platform, we call that our PEC Direct, again, requires immune suppression today, but tomorrow with our immune evasive cells may not. This direct vascularization approach has shown patients producing C-peptide, and that is a biomarker for insulin. These patients, when they were implanted with our device, were showing no C-peptide. So de novo C-peptide production, improved time and range trends, evidence of histological evidence of cell engraftment. And we've continued to optimize along the way, from surgical procedures, the number of devices, where to place them, and importantly, working with CRISPR to develop the immune evasive cell line, PEC-QT. On the right, we have innovated with Gore to use a membrane, again, that requires or enables surface diffusion. This would be an encapsulated device. Again, this would be not needing immune suppression when we use this device in our clinical trials. And we're optimistic that this device will also provide therapeutic benefits for patients. Just to give you an example, a snapshot into the possibilities on the open device, just taking one of the patients. So this is really just an N of one, but give you a sense of where we can go with the technology. On the left here, you can see staining from an explanted device that we can actually see what's going on in the device. We saw evidence, as you can see, from these brown clusters of insulin cells being produced, and on the bottom, glucagon cells. So that shows that the cells are engrafting. That's that early histological tracking that we do. In the right, you can see on the right, the level of engraftment now is mature enough to produce C-peptide, a biomarker of insulin. All these patients were C-peptide negative at enrollment, indicating that they were not producing insulin. And across the spectrum, you can see there's a spectrum depending on how many devices in the patient's response. If we take just one of those patients that are exemplary and kind of track that along the timeline, you can see here the kind of clinical parameters that could be impacted. Here you can see a patient that had a meaningful improvement in their average glucose and meaningful reductions in the high glucose levels and low glucose levels, hyper and hypoglycemia. But importantly, and I've highlighted here for you, the time and range, which is a very important parameter. It's a byproduct of the amount of time you've minimized high and lows, obviously. But to get to 74%, that's a very high and acceptable range for a patient to be in, and a diabetic patient. And as on the right, you can see what that equates to is a significant more time, 22% improvement in their euglycemic range, and the impact downstream on long-term complications that could have is very meaningful. As I mentioned, we are looking to optimize our devices, looking to continuously improve to reach the goal of providing a functional cure for type 1 diabetics. And in this case, we enable that mission with the help of two partners. CRISPR is a worldwide leader in gene editing. Regenerative medicine is one of their most important strategic pillars. And our program, where we make gene edits to help our cells become immune evasive, is a critical program for CRISPR. I'll have more to say about that in a few slides, but it's been a great partnership, and really the benefit of using the benefit of evolving science to hopefully have a situation where, again, patients do not need to take immune suppressant drugs. On the right, we have had a long-term partnership with Gore. Gore is the maker, as you all may know, of Gore-Tex. They have a very substantial and robust biopharmaceutical division that we partner with. They're a leading membrane science, material sciences company. The technology here we use to use in our encapsulated device, again, requires or optimizes on surface diffusion, but really that's to mitigate the foreign body and giant cell response that we get from patients on implantation. So we're looking for very specific and proprietary permeability and diffusion criteria as we look for the innovation on these devices. Just a note on the landscape, as you can see here across the various companies, really ViaCyte is really in the lead on a couple of fronts. We've taken our programs from bench research to bedside research, and our open and closed device platforms are in clinical trials in patients. We are, as I mentioned, in the IND enabling phase with our QT CRISPR collaboration cells, and we are also exploring naked cell insertion or application vis-a-vis the portal vein. So we have multiple programs in advanced stages across the spectrum of possibilities to improve outcomes here. No other company has a spectrum of these clinical approaches in their portfolio. So here's a snapshot of where we stand on our current clinical programs, and I'll start at the bottom. The PEC NCAP using the Gore membrane. This is an encapsulated approach. We started this trial in January. Our goal was to get 10 to 15 patients. I'm pleased to report today that we've enrolled our final patient just a few weeks ago in early May. And so we'll be waiting for that full group to report out on 6 months C-peptide and time and range data by the fourth quarter of this year. So we'll watch those patients as they progress. On the top, we're very excited about the open device platform. We had intended to enroll 10 patients here with the open approach requires immune suppression. We did not get, we got implicated or hampered a little bit with COVID. I'm pleased to report that we're having good site visits, and we anticipate being able to be fully enrolled in our trial here in about the end of July, early August. There we will report C-peptide and time and range as well, and that data now will be available in the first half of 2022. Additionally, we're currently fast at work. We're working with CRISPR to develop the seed banks and the protocols ready to be submitting that to regulatory authorities and have that in humans by the end of this year. And we'll have a readout of the cell immune evasion and safety properties in the fourth quarter of this year, maybe early first quarter 22 for the full components, and then moving first in human efficacy trial in the middle part of 2022. So these are the big catalysts for the company with our current programs. And please, and just want to say thanks to the team for their ability to stay focused and execute on these during a pandemic. As many, I'm sure, folks have been reporting, that's not a trivial thing to do, and really the dedication of our employees should be recognized. As you can see here, this really talks about our approach, and looking on the left, you can see the discs that represent those cell stages I described earlier. We start with a pluripotent stem cell. Today, we differentiate, across the spectrum of cells to the pancreas, but we could branch off and really do anything in that, 2 o'clock to 4 o'clock or 1 o'clock to 4 o'clock range. Today, we're exploring other areas to differentiate with and explore other disease areas that are, low cell dose, high therapeutic index, immune privileged kind of opportunities. So we'll be describing those more as time goes on. But it's important to recognize that we have the technology and the expertise to do so, and more importantly, we have the IP to do so. We have a very, very robust IP position, well over close to 800 patents across multiple types of descriptions, how to differentiate and scale cells, what they become, pancreatic endoderm as an example, or what they do. And then on the left, patents covering the combination and the manufacturing and the design of these membranes. So we have a matrix set of patents, and they go out to the end of the 2030s. So very robust freedom to operate here at ViaCyte. Importantly, we've made consistent and considerable investments in our ability to manufacture. We control our own manufacturing. We've invested and have the ability to make both cells, devices, and combination products. They've been regulatorily tested, and they have been consistently produced under GMP level criteria. And I just highlight there on the left that no competitor has manufactured and implanted in humans any pluripotent stem cell derived GMP level product in the diabetic patient. So we're blazing trails here, but we believe this is a fundamental capability that we have that we want to continue to nurture, invest, and expand. And then finally, you have to get this all done with people. We're blessed to have a team at ViaCyte that is a mix of both foundational scientists and people who've been at the company for some time. You think about a Liz Bowie or Kevin, and we brought in a substantial amount of new talent to kind of keep the talent and mix fresh. Steve White recently, Brittany and I have all joined the company of late. On the right, you can see we have a good board of directors from a wide variety of different investment and company perspectives, and they're very engaged and open to our ability to ask them questions, and we meet with them often. So the combination of both the management team and the expertise of the board of directors allows us to really navigate the future in a very, I think, efficient manner. So with that, I'll just pause, say thank you to the folks here at Jefferies to allow us to share our story, and hopefully there'll be future interactions with all of you in the future.
Great. Thank you, Michael, for the wonderful presentation. I think it looks like we have a couple minutes for questions, and we have a couple questions from the audience, if that's all right.
Yeah, that's great.
So the first question was, can you provide any clinical trial data for the PEC NCAP?
Right. So the PEC NCAP, let me give you a little bit more details there. It's a multi-site study. We have 14 patients enrolled. It is open label. We have not received any what I would call foundational data. So what I mean by that is we need a patient it's thought that the patient has to reach the 6-month mark before we can start to see meaningful signals of engraftment. And at this point, we haven't quite gotten anybody to 6 months. So I don't have anything to report there except to say patients are doing well. I actually went and witnessed one of the implantations. It's an amazing procedure. But at this point, there's nothing clinically to report on NCAP, but we will report on data that we received. I would say on the open device, one thing I would say is on the PEC Direct, we had 2 patients that went into clinical trial prior to COVID, and we will report on some clinical data at the upcoming ADA on that device, on that program.
Great, thank you. And then as far as the competitive overview and the competitive landscape, can you comment on how you think about Sernova?
Right. I think that there's different technologies and approaches, and one of the approaches that you can use is microencapsulation. The other we've used is device encapsulation, and another approach you've heard companies talk about, portal vein, which is mirroring kind of the cadaver islet cell transplant. We've decided to go into the direction that we've pursued with devices. The devices that we have, I think, have some regulatory affinity because they can be explanted, so the safety question becomes a little more digestible by the regulatory authorities. So I think that that's the thing that I would comment on is there's two principal things. One is the cell types, and the other is the types of encapsulation or device approach. So those are just the variabilities in kind of the clinical approaches people are taking.
Got it. Thank you. And then maybe we have time for one more question here. So do you still anticipate the filing of biologics license applications in 2024 for PEC NCAP and 2025 for PEC Direct?
Those are definitely in the range of possibilities for sure. One range of possibility would be, for an example, on the PEC Direct program, if we see seminal results, and really for both of these, if we see seminal results on these drugs, on these cohorts, we would probably file for a RMAT designation, which would allow us to have a more of a fast track type of approach with the agency and show meaningful benefit. I think one thing to keep in mind, while I mentioned that our goal is to get to a functional cure, and that is our goal, the first set of products we come out will be probably meaningful instead of functional, in the sense that we may not disintermediate completely insulin, but will provide enough benefit to the patients to minimize insulin and improve time and range and have clinical outcomes. So our first set of products will probably be meaningful clinical impact products, which would meet the definition of an RMAT designation.
Great. Thank you, Michael. Well, I think that's it for time. So I really appreciate you spending some time with us this afternoon to give the presentation and answer some of our questions.
Caleb, thanks. And thanks to all for listening. Appreciate it. I'm happy to take questions later on.